In the first session, our first speaker for the day, Professor and Director of the Nutrition Science and Food Chemistry Lab at Virginia State University, Petersburg, Virginia, US. Dr. Siddiqui has earned his BS and MS degrees in biochemistry from the University of Karachi in Karachi, Pakistan. He obtained his PhD from the Australian National University in Canberra, Australia. At present, Dr. Siddiqui's research interests are in the area of cancer, inflammation, and cardiovascular diseases by a combination of nutrients and drugs. He received.
Sorry. So the topic of my presentation is ginger extract modulates interferon 1 signaling pathway in hepatocellular carcinoma cells derived from Caucasian, Asian, and African American patients.
Thank you so much, Dr. Kaswani. Moving forward, I request our chair, Erika Yasuhara, to introduce our next speaker, Professor Khalid Shah.
Okay. Professor Khalid Shah is the Vice Chair of Research for the Department of Neurosurgery at BWH and Professor at Harvard Medical School. He directs the Center for STEM and Translational Immunotherapy and also Joint Center of Excellence in Biomedicine. He is also the principal faculty at Harvard Stem Cell Institute in Boston. Professor Khalid Shah and his team have pioneered major developments in the stem cell therapy field, successfully developing the experimental models to understand basic cancer biology and therapeutic stem cells for cancer. These studies have been published in a number of very high-impact journals, like Nature Neuroscience, PNAS, Nature Reviews Cancer, JNCI Stem Cells, and Lancet Oncology. Professor Khalid Shah's stem cell work has caught the attention in the public domain, and as such, it has been highlighted in the media worldwide, including features on BBC and CNN. Recently, Professor Khalid Shah's laboratory has reverse engineered cancer cells using CRISPR or Cas9 technology and utilized them as therapeutics to treat cancer. This work was published in journals Science Translation Medicine and highlighted worldwide, including features on Scientific American, New York Times, and NPR. Amongst Professor Khalid Shah's published works are 2 books featuring groundbreaking insights into stem cell therapies for cancer, stem cell therapeutics for cancer, and mesenchymal stem cells in cancer therapy. Potential developing novel cancer therapies by him and his team have been recognized by many cancer alliances and associations. Based on this, he has received the Young Investigator Award from Alliance for Cancer Gene Therapy, Research Fellow Award from the American Cancer Society, Distinguished Research Award from Academy of Radiology and Innovation Awards from James McDonald Foundation, American Brain Tumor Association, and Goldhirsch Foundation. Recently, he has received Department of Defense Idea and Impact Awards for his work on treating metastatic brain tumors. Professor Shah has often been invited to serve as a grant receiver for the NIH and has reviewed applications for granting agencies in the US, Australia, and United Kingdom, such as the European Science.
Change it basically in social change. It's called education. It happens in political scenarios, it happens in different cultures. And then the people who agitate, they want to bring the change, and then people sort of in the society walk with them and say, okay, let's make the change. So that's where innovation happens. When you're not happy with status quo, you want to bring the change. And innovation is there as quo. You want to change it basically in social change. It's called education. It happens in political scenarios, it happens in different cultures. And then the people who agitate, they want to bring the change, and then people sort of in the society walk with them and say, okay, let's make the change. So that's where innovation happens. When you're not happy with status quo, you want to bring the change. And innovation is there, but if it's not orchestrated, if it's not really sort of implemented, it will not become a change. It will stay as an idea. It will not go to the next level. And that's where the orchestrators are needed. And in medicine, it's the same thing. That if you innovate and if you don't have people to orchestrate it and take it to next level, it doesn't mean anything. So bringing it from social concept to science and to medicine is sort of is critical. And you need sometimes an innovator. It's not the same as orchestrator. You know, it could be somebody else. It could be a team that he has given or she has given his innovation to them and say, okay, take it to clinics. And that happens in academia. You innovate and then you either start up companies or you work with other companies so that they have money and they take it to next level. And again, impact requires all three. Now, I'll give you a few examples. We always had this concept that if there's a seed, we plant the seed in the soil, you will get the plant, and the plant will have the leaves. The leaves will never become the seed. You know, this is all of us know that it's very hard for a leaf to become a seed. So in medicine, if you have a fully differentiated cell, like a skin cell, it's very hard for it to become a stem cell. Originally, it came from the stem cell. It's very hard for it to become a stem cell. But so we were limited with stem cells because we had to derive these from embryos. This is late 1990s, early 2000. During Bush era, you might have heard embryonic stem cells for ethical reasons were not allowed because, you know, they had to abort the embryos and stuff. So somewhere, and that's why I say that somewhere somebody was innovating. And in 2008, Shinya Yamanaka got the Nobel Prize for saying that now I can convert this leaf into a seed. He didn't do it with plants, but he did it with animals or
sensitivity, insulin sensitivity. It also has several endocrinal functions that are poorly understood now. So due to its effect on the glycemia, triglyceride clearance, and insulin sensitivity, it's of particular interest for patients that suffer of diabetes, obesity, and metabolic complications. Because brown adipose tissue, called BAT, is using glucose and triglycerides to create energy and dissipate this energy into heat instead of stocking it through, for example, ATP like other tissues. So I'm going to quickly speak about the developmental trajectories of brown adipose tissue. Why is it important to us? Because our main goal is to synthesize the tissue in vitro. But to synthesize this tissue in vitro, we need to fully understand what's going on in vivo, how the tissue, the stem cells are giving rise to a full mature brown adipose tissue. And then we try to imitate life. So this is an embryo, 3 different steps. And we will give interest, especially for the last one, the gastrula. The gastrula is the step where the embryo is composed of 3 layers, the ectoderm, mesoderm, and endoderm. So the mesoderm, the layer we are interested in, will give rise to several tissues, including muscle, cartilage, bones, and brown adipose tissue. So with time, the mesoderm will itself segregate into different parts. The PM, the paraaxial mesoderm, which is in both sides, lining in both sides of the neural tube after its formation. There is the intermediate mesoderm, EM, and the LPM, the lateral plate mesoderm. So today we'll focus mainly on the paraxial mesoderm, because the paraxial mesoderm will segment it itself through time from in the anterior posterior gradient, and will create somites. And these somites, which are like small spheres in both sides of the neural tube, will give rise to several cell types, including the brown fat, skeletal muscle, bone, cartilage. So if we look at the fat lineages, because there is different type of fat, the brown fat we are interested in, there is also the white fat, the fat we all know that is used to stock triglycerides, our energy reserve. There is also pink fat, which is mainly for lactation, for pregnancy, and beige adipocyte, which has a strange phenotype between the brown and the white adipocytes. So in this figure, we can see that there is two types of lineages, the MIF negative progenitors and the MIF5 positive progenitors. The MIF5 negative progenitors is mainly dependent of the lateral plate mesoderm trajectory and will give rise to most of the fat lineages. But the MIF5 positive progenitors will give rise to both brown adipose tissue and skeletal muscle tissue. And both of them are coming from the paraxial mesoderm and the somites trajectory. So our main goal is to try to reproduce this in vivo. And as I said, to fully being able to fully recover and reconstruct the brown adipose tissue in vivo for example, cell therapies. Let's say if we are able to synthesize this kind of cells and we are able to graft them in patients that are suffering from obesity, diabetes, or metabolic disorders, we can improve their global metabolism and help them recover their phenotype. So the main strategy nowadays to understand how a trajectory is constructed from the stem cell to the mature tissue is to conduct transcriptomic analysis through the single cell RNA sequencing that have been already introduced by Professor Siddiqui. So we conducted this on mouse. We dissected the neck part of a mouse embryo from 11.5 days post-fertilization to 15.5 days post-fertilization. As I said, neck is one of the main depots of brown adipose tissue. That's why we focused on this region. And then we analyzed it computationally and got a UMAP, which is a mathematical representation, dimension reduction, that allows us to visualize really easily if two cells have really close transcriptomic profiles or not. It allows us to clusterize the cells and knows according to their profile, according to the gene they express, whether they are brown fat, muscle, cartilage, or whatever. So this analysis allowed us to identify several checkpoints really important. For example, the mesoderm fate is mainly expressed with PAX3, which is the main marker for the mesoderm. And the PAX3 populations will give rise to PAX7 populations that are the same as the MIF5 positive that I showed before. So these PAX7 MIF5 positive cells are progenitors that will give rise either to brown adipose tissue or to skeletal muscle. If the population continue to express PAX7, then we will get skeletal muscle. But if it stops expressing PAX7, then we will have a pool of a different population that we call fibroblastic progenitors that can give rise to a lot of tissues. And among all the population that derive from the fibroblastic progenitor,
this wave-like form when I increased the shutter speed. And when I took a picture with the high-speed camera, I found that the actual movement of the string was just vibrating up and down. And the undulating waveform was a apparent phenomenon caused only by an old camera. And just for a fact, the iPhone 5 I used had a 30 frame per second speed. So the results here, as you can see, on the left is the iPhone 5 results, showing a undulating waveform, which is showing like a sine kind of waveform. And then on the right is the actual movement of the string, which is just going up and down. So based off of this, we could see these two types of cameras, the rolling shutter camera and the global shutter camera. And older iPhones, as I explained before, use a shooting method called rolling shutter. And when recording an image on the camera sensor, they slowly move the vertical line from left to right. So in other words, there's a time difference between the left side and the right side of the screen when shooting. And even during this time difference, the actual violin string continues to vibrate up and down many times. So the image captured in the camera's memory creates a waveform. However, with the newer phones, the one screen is shot at the same time, so there's no time lag like in the older phones, so you can't see the waveform, which shows the actual up and down movement of the strings. And then I calculated the wavelengths of these waveforms in the iPhone 5, and that gave me that the string vibrates 5.31 times during this 1/30th of a second, and this can be used to calculate the frequency of the violin. So based off of my first experiment, I could tell the relationship between waves and strings, and this led me to become interested in the fact that the camera was able to record waveforms that were supposed to be invisible to the naked eye. And after thinking about it, I realized that the ability to visualize sounds has many applications. And for example, I thought that being able to see the movement of a string waves like this for people with hearing impairments would be useful in providing new information. And this leads me into the second experiment, where I thought of the ability to visualize sound and if it would be possible to visualize sound waves propagating through the air. So I conducted an experiment based on how waves can travel through the air and how they cause the air to vibrate. So to visualize the vibrations in the air, I first glued a transparent cylindrical bead onto a glass slide with a glue gun, and then using a candle, I smoked the cylindrical bead, and then I set it under a microscope and observed the movement of smoke particles through a microscope by illuminating the smoke from the side with a green laser pointer. And then this resulted in the fine movement of smoke particles known as the Brownian motion, and this was observed even when no action was taken. So smoke microscopy was found to be the most suitable experimental system for observing the fine movements of particles. And the pattern of Brownian motion of smoke particles changed slightly when a violin string was struck. So it turns out that the vibration of smoke particles is affected by the sound waves of the violin traveling through the air. So as you can see here, that smoke particles are what's glittering in this image right here. And they are the little dots that are shown glittering and conducting a Brownian motion. And depending on the sound of the violin, the pattern of the Brownian motion changes slightly. And as you can see here, the Brownian motion is a really random-like motion that happened whenever the violin string was stuck. And this leads me into my third experiment, where how the human ear picks up the sounds from the air. And
Hi, thank you for the introduction, and I just wanted to say thank you for this opportunity. So today I'm going to be talking about identifying the risks of COVID-19 infections and mortality using a computational approach based on the genetic background of the patient. So just a little bit background. As we all know, coronavirus is an infectious disease caused by the SARS-CoV-2 virus. The CDC and the WHO had previously declared this virus to be a public health emergency of international concern. And this virus has had a rapid increase, as we saw, And as of now, there are about 772 million confirmed cases and about 6.9 million deaths across the globe. Research shows that the cause and predictors proved crucial in affecting the outcome of the virus. Our research explores age and the pre-existing conditions based off of the COVID-19 mortality rates. This presentation will delve into these knowledge gaps and propose an approach to address these critical issues. So, grasping the pre-existing conditions linked to COVID-19 mortality is crucial for shaping our response to the pandemic. By identifying the individuals who are at a higher risk of severe outcomes, we can prioritize their preventative measures and targeted therapies. Additionally, it enables tailored public health interventions and treatments, making our pandemic management more effective. Understanding the underlying factors behind the COVID-19 mortality fosters a more proactive approach where we can personalize medicine and offer the best strategies to help reduce the risk. So our goal for this is to comprehend the pre-existing conditions and to help us predict what outcomes may come from COVID-19 virus. So we took a comprehensive approach in examining these pre-existing conditions and the age factors associated with the immune response to the COVID-19 virus. This perspective does not only identify the patterns, but it also is important in shaping precise and individualized treatments and preventative care. This personalized medicine and public health strategies helps to optimize the outcomes for individuals at a higher risk for fatality. So our approach involved carefully collecting information to better understand COVID-19 mortality. We used computational techniques and Python tools to analyze the data to explore how pre-existing health conditions, including influenza and pneumonia, hypertensive disease, diabetes, and Alzheimer's, and how they relate to COVID-19 deaths. We also looked at the mortality rates from different age groups from 2020 to 2023. So this data was pulled from the CDC public database, specifically on the mortality rates in the United States. And this data was collected from about 1.1 million people with pre-existing conditions. And this practical approach helped us uncover the interplay between age, health conditions, and the risk for mortality. So using the CDC database, we used a computational program to classify the website were plugged into a Python program that I created, which helped us to curate the data graphs to show the results. So our first graph is on the pre-existing conditions. So the highest one we can see is from influenza and pneumonia. We can see a drastic difference in this disease. And we can see as the higher age someone gets, the higher risk or higher fatality rate there is, specifically ranging from ages 65 and higher. So drawing insights from our data analysis, we observed a significant disparity in mortality among pre-existing conditions such as influenza and pneumonia. This indicated an elevated risk of fatality for this specific demographic. And our findings also show progressive change in the mortality risk increasing with age, with the highest rates observed in 75 to 84 age group. These patterns are precautions, particularly individuals in this age bracket in these conditions. This will help us to mitigate the risk of severe outcomes. So moving forward, my plans in the future involve
and its benefits universally, the scientific method and AI's ability to enhance it, specific ways of learning resource management, and finally, specific things we can do to encourage AI development while still being safe. I don't want an AI takeover. Why does the climate change really happen? Well, every year, humans use fossil fuels to drive their cars, and even digging up a lot of lithium batteries for electric cars takes up a lot of fossil fuel. And also the natural production of greenhouse gases by the Earth itself all gets trapped in the atmosphere, which causes the heat to be like a car on a hot day, which is much hotter than its surroundings. Next, let's talk about prevention versus protection. There are two ways to deal with climate change. Firstly is prevention, and the second way is to adapt and protect while it's happening. Think of it like treating a disease versus preventing the disease. And in this situation, if it's as bad as cancer, which my dad can tell you is very difficult to treat, we need to prevent climate change, not protect against it when it's already here. To do this, we need to manufacture new low carbon tech that can capture carbon and utilize more efficient energy systems. But we have some issues. We need to make sure that this is not toxic to humans. We need to be as resource efficient as possible so we can limit the amount of carbon being released from creating these new compounds. And finally, we need a lot of money. A brief introduction to AI and its universal benefits. Imagine you are someone with photographic memory. You can remember anything you've ever seen, heard, done. You can remember anything. Word for word, bar for bar, letter for letter. The kind of things you can do with this ability are absolutely endless. You could imagine if you also store more information than anyone in this building and on the Zoom call can imagine. But that is now a reality, as AI is really the future, and we will universally use it to improve on life. Because you had the ability to ask someone with photographic memory any question, and especially one that had every piece of human knowledge in existence. How would you use it? The scientific method and how AI can improve it substantially. We've all seen the scientific method. We have your question, your hypothesis, your experiment, analysis, conclusion, and then you start with another observation. AI can help with questions because it can take huge databases and generate new questions that we can then take into labs and be confident that these questions will not be taken. Secondly, it can help with hypothesis because the AI can help predict what is going to happen using machine learning and again, a very, very wide database. Thirdly, experimentation. AI can again experiment as fast as possible, and this is almost instantly and much faster than a chemist and somewhat automate creating compounds. The ideas of database, machine learning, predictive capabilities, and automations will help us make science more efficient in general and also help solve climate change. Machine learning and resource management. A big issue when tackling climate change is making sure that every resource that we use to either test ways to prevent or actually prevent climate change is not going to be used is not going to be wasted. And we can use machine learning. Machine learning is just a machine learning how a human would. However, this happens much faster. This fast learning will lead to unparalleled levels of intelligence that will make dealing with resource management much easier, such as solar and nuclear energy that can be used more effectively to give energy to us in better ways. Specific things we can do to encourage AI development.
Adipose tissues as treatment for obesity and mellitus diabetes.
And so first of all, we are going to Mariam.
Oh yeah, you can just use the arrow through that, yes. Yes.
Okay, so we're gonna have Dr. Shadi, we're gonna have Dr. Ashraf.
Good afternoon, everyone. I would like to thank the organizers for the invitation to present my work. My topic for today's talk is what are the genetic and environmental factors that lead to Fuchs' endothelial polypoidal dystrophy. So I have been basically working on corneal diseases, and the most common disease I work on is Fuchs endothelial corneal dystrophy, which is a genetically complex disorder, and it is very much prevalent in females. So I would say of all the patients we see with FECD, about 70% of those patients are females, and nobody knows why females get it. Not even today. So much research has been done genetically and also looking at the environmental factors, but this question still remains the same. So that makes it really interesting. It is age-related degenerative disorder, and the clinical characteristics of this disease are the loss of corneal endothelial cells, sorry about that. All right, so the clinical characteristics of FECD include the loss of corneal endothelial cells, which include large cells. And then we see clusters of cells on the cornea of the eye, which we typically call guttae, corneal edema, and there is also loss of vision. So if we talk about the genetic causes of FECD that are known so far, we first need to know that we can actually divide FECD into two different categories. One category is the early onset FECD. This early onset FECD is very, very rare. Usually, we don't find it. It's like 10% of all the cases known in the entire world. And it manifests early, like it manifests in the first decade of life. So decade is like first 10 years of life. So, just it being very rare, to get samples of such patients get even more difficult. And to do genetic analysis of those samples get difficult, as obvious. So, so far there had been only one gene discovered where mutations can cause this early onset FECD. It's called collagen 8 alpha 2 gene. And then there is this common category of FECD, which we call the late onset of FECD. So it's more typical. And this manifests in the second or third decade of life. And even like, the more symptoms are seen in the fifth decade, which is like plus 50 years of age. So most of the studies that we know that have been done for patients in FECD are now done in the late onset FECD, and that's why we see more literature and more research on that. And there have been genes discovered as a cause of late onset FECD, and some of the common genes are like transcription factors like TCF4. There are also transcription factors like ZEP1, and then several others. There are also some locus that have been discovered, but there are not definitive genes that we found there in that locus. So, we don't know. And then what is the mode of inheritance? So the mode of inheritance would be dominant, which means that one single heterozygous allele went from the parents to the kids is sufficient to cause the disease. So the most common, I would say about 70% of the cases of FECD, which have been genetically analyzed, are caused by mutations in a gene called TCF4 or the transcription factor 4. Transcription factor 4 encodes for helix loop helix, transcription factor E22. And there had been association with a SNP that if it finds the SNP in the patients, they might have the disease, but the most common mutation identified is actually
derangements, which includes muscle wasting and systemic inflammation, which is what we focused on in our research. So usually muscle cells are known as motor cells or metabolic cells, but they're also endocrine cells, which means that they will excrete a chemical or a hormone. And here we have a list of cytokines that are excreted by muscle cells. And you can see that the excretion of these cytokines has an effect on a lot of processes, including the muscle. Some of the processes listed are it affects muscle mass or muscle atrophy or muscle regeneration. So our hypothesis was that when there's no inflammation in muscles, the muscle mass is maintained and mitochondria is healthy. And the inflammatory response is also, there's a good balance between the pro-inflammatory response and the anti-inflammatory response. And then in contrast, when there is muscle inflammation, that can cause muscle mass loss, mitochondrial defects, and then there's also a disruption to the balance between the inflammatory response. So the methods that we used is that we created muscle cells using the C2C12 cells, and we transfected them with plasmids, including NF-kappa B and ROSA 26 CRISPR-Cas9. And then we took these cells and ran them through the FACS to make sure that we had cells with rich GFP expression. And then we tested these cells in the presence of burn serum and LPS, which are both inflammatory, induce an inflammatory response. And then we tested these drugs in the burn serum and LPS with the two different drugs. We had Trehalose and Humanin. Trehalose is a drug that has been shown to increase the beneficial effects of autophagy and mitophagy. And then Humanin has been shown to help with Alzheimer's disease. So in our first round of results, we used the LPS. And the first image here that we have on the left is just the cells without any LPS, without any of the drugs. The second image is the cells in the presence of LPS, and you can see that there is much higher fluorescence in the cells, meaning GFP was expressed more, and shows that there was more mitophagy and inflammation. And then we have the LPS in the presence of Trehalose. You can see that from the second to third image, there's a big decrease in the amount of fluorescence and inflammation. And then similarly from the second to the fourth image, which is in the presence of Humanin, there's also a decrease in the fluorescence. And it's also important to note that all of these images were taken in similar exposure conditions. So here's a graph of the data that I just showed you, and it quantifies the GFP fluorescence. And you can see that from the LPS to the control to the other drugs, there is a significant decrease in the GFP expression. And then we repeated the same thing in the presence of burn serum instead of LPS. And then very similar effects. You can see that the burn serum increased the GFP expression.
So what we are working here to understand the first physiology and how during sedentary lifestyle we can counter the development of insulin resistance and obesity. So maybe some of you are familiar with the, how we are human develop diabetes, and for the youngster they are not familiar with how we can understand the diabetes. So there are several factors for the cause of a type 2 diabetes. So predominant factor is environmental factors. So that contributes to more than 90% for the development of insulin resistance for type 2 diabetes. And genetic factors contributes to 5 to 10% in the patients who develop type 2 diabetes. If I go on left side, that in sedentary lifestyle, obesity. In obesity, basically central obesity, like visceral fat increases in the patients. And there are a lot of many mechanisms involved in the increased like visceral fat in these obese patients. And ultimately, it develop into insulin resistance. And meanwhile, the pancreas also is not sufficient to cope that inflammation or that insulin resistance. The relative deficiency of insulin also occur. So it result in whether decreased insulin action or the development of insulin resistance. And ultimately, it goes to type 2 diabetes. So in the development of type 2 diabetes, the three metabolic target tissue are mainly involved. Liver is the main organ, like to produce glucose, and muscle and fat cells are the main consumer where glucose can be consumed, or we can say glucose can be utilized from blood to the cells. Any defects in muscle and adipose tissue to decrease the glucose uptake or utilization results in hyperglycemia, or in other mechanism, liver produces more glucose and our body is not able to take up that one. So ultimately, it results into type 2 diabetes. So my PhD work, I was mainly focusing on adipose tissue. And in second project, I have done little bit like research on skeletal muscle, how the muscle is important. So as some of you are familiar or not, so I would like to say, in past, fat cells or adipose tissue was known as an inert energy storage organ. And current, we have know that adipose tissue is not only a energy storage organ, but it possesses many different kind of cells, and that cells can interact with each other, and that can contribute systemic metabolism. And we don't know in future what is going to happen, like because we are working on adipose tissue, like white adipose tissue, brown adipose tissue, as Dr. Yasura was talking about, like mitochondrial biogenesis or mitochondrial energy storage house, brown fat. So these are all like still we are investigating how can adipose tissue can contribute to systemic metabolism. Here I would like to say like what's in adipose tissue. Adipose tissue comprises of stem cells, fat cells, progenitor cells, fibroblast, pre-adipocytes, and recently discovered like exosomes or microRNA, that fat cell can produce these exosomes and that can travel to the blood circulation and it can target other tissues. And immune cell, there are several immune cells, T cell, B cell, other immune cells, but our focus was mainly macrophages. Macrophages could be beneficial, macrophages could be detrimental. So the macrophages that have properties to enhance the inflammation or pro-inflammatory cytokines that we called as M1 macrophages, and the macrophages that can counteract the inflammation are known as anti-inflammatory macrophages. What happens in fat or adipose tissue, all these cells communicate and interact with each other, and it can transmit signal to other organ or tissue as well. Hence, they can regulate various physiological process, including glucose metabolism. So based on my previous background, so we have done several studies on immune cells in obesity to find out the interaction, how immune cells can regulate progenitor stem cells. So today I would like to discuss only two studies in which I have found how M2-like macrophages or anti-inflammatory macrophages can regulate systemic glucose metabolism. And in second part of my talk, I will little bit talk about, diabetes. When the patients who have developed diabetes, they are more prone to if they have injury, their regeneration become very slow, and how we can improve their injury if they develop diabetes as well as injury. So first, what happened in obesity? This is previous studies even. So some studies in our lab, we have discovered that in lean state, there are large number of anti-inflammatory macrophages in white adipose tissue. When the mice become obese, there is infiltration of macrophages in the adipocytes, and these infiltration of macrophages ultimately cause insulin resistance. I was first interested to know why these like lean mice adipose tissue have more M2-like macrophages. And yes, these macrophages are required for adipose tissue like remodeling or in the regulation of stem cells. So I will try to focus like what kind of M2-like macrophages play a role. What is known so far, M2-like macrophages promotes insulin sensitivity. There are several mechanisms. I will not go in deep. Impairment in the number or functions of M2-like macrophages can result in insulin resistance. And M2-like macrophages promotes browning of white adipose tissue upon cold stimulation. Even though
you guys, most of you perceive, I'm an interventional radiologist, so I operate day in, day out, 99% of the time. That 1% is because every now and then somebody wants me to look at an image for a diagnostic purpose. That's when I say that. Otherwise, I'm a minimally invasive surgeon and I mostly deal with cancer. So in a nutshell, so I'll give you the historical perspective because this talk is designed to excite the young generation to see what we can do, what's possible, so that they can come join us and push the future. So I'll give you a historical background. So vascular interventional radiology, this is what my full field's name is. What do we do? So the second paragraph summarizes it well, like we open things, we close things, we put things in, we take things out through vascular and direct puncture access. And that's pretty much it. And we use a lot of novel devices, tools, and techniques, which again is very well versed for our field and our residents and fellows. But for you, it might be a little bit novel. So I'll keep showing you a little bit, and eventually I'll show you some examples also so that you have some sense of what I'm talking about. So historical perspective, if you talk, like we are collaborating with surgery, we are technically seen as surgeons. But surgeons have a long history. The longest reported data of surgery is from the Egyptian mummies. There was one of the mummies who had a mastectomy. So that's like probably 3, 5, 7, anybody guess 1,000 years old. For us, like, as I said, I come from radiology. There was nothing before 1896. One curious instance laid the foundation of this whole field, and now we are like so much invested that I can challenge any one of you, go through the hospital's alleys and not see us. Good luck. So in 1896, Wilhelm Röntgen found this, and then soon we started thinking, like, hey, what can be done? What can be it used to be? So then they started finding contrast, and in 1929 we found contrast, and that's when IR started being, because we can see things, started seeing things. And all these things took some time. By the time it got to refine to a certain point that we can use it in a clinical standpoint, because as we know, most of us here are researchers. It takes a lot before anything goes from the bench to the patient. So it took like at least a half a century before we could do something. So even punctures were like very rough in those days. So in 50s and 60s, there was a guy in Sweden, Seldinger, Gwen Swan Seldinger, Ivan, and he was in the Karolinska Institute. And he came up with a brilliant idea that you can have a single puncture of the wall and you can get to the vessel. And from there on, it was no stopping us back. We were the first one who did all the cath lab works, all the cardiology imaging till almost 1980. And then cardiology said, oh, enough. Like you guys just do the procedure. We see the patient. So why don't we do both? And they learned from us and they took over. Rightly so. I'm not complaining, but we are the innovators basically. Then fluoroscopy came. And then in 1964 was a very pivotal year for us, the way we practice now. And I'll spend a couple of slides talking about that, because that's when Charles Dotter, who's kind of termed as the father of IR in the US. He was from Long Island guy, but he came to Boston for practice, eventually went all over the country and eventually put his anchor in Oregon. So that's why the institute there is known as Dotter Institute. So he started doing this. So as I was saying that we were started imaging, surgeons will send the patient to us and they will specifically mention in the note, just image, don't do anything. But you know, it's very hard to stop an inquisitive guy. He started looking at it closely, and what he did was, like, in one of the patients, he was putting a catheter. It was a thigh, like we call it SFA stenosis. He was injecting and his catheter moved by mistake. When he pulled back, the narrowing opened up. And that opening up, and these patients come with ulcer of the foot. That ulcer started healing. The surgeon was mad. Like, why did you do that? They're like, oops. But next, I was like, oh, now I know what to do. What he started doing, he will pass smaller catheter, then bigger catheter, and then much bigger catheter. And that used to be termed as daughterization, which is now replaced by balloon angioplasty, which I'm sure most of you have known. Somebody in your surrounding must have had a coronary cath with an angioplasty, which is the most common way you hear about it. So daughter led that base. And this is what I was showing, like the image. And this is the first image of IR. And you can see, I don't know if my cursor is visible to you, but you can see there's a narrowing right at the knee bone in the first image on the left. There's a slight narrowing, and that was opened up, and this is gangrene healed. It took some time, and then eventually in 1974, Grunzig was another guy, a German root guy, but he came to US. He had like almost 200 patents. He was like machine, and he died at 47, very young, but he was so brilliant that he could do anything over a weekend. Again, so with this, we kept moving on. We start looking at different organs. So again, there's a very one basic conceptual difference between us and the rest of the physicians. Any of the physician you talk to, they're married to an organ system or a disease system. We are